We're all set, right?
That's good.
Okay, great. So hello everyone. My name is Noushin. I'm working calling Belta from Boston University, and we are actually in robotics lab. So I guess our work is kind of different, but also at the same time related to what other people are doing. We are more involved in providing automated frameworks for designing spatial or spatial temporal patterns. And oops, there are some problems. Basically, the reason or like the motivation of the work that we are doing is that there is not enough systematic approaches to design organizations with some tunable parameters. So imagine you have a model of the cells or like the system that you are working with, and you want to like achieve some specific behavior within that systems, but definitely doing it with trial and error or like through the experiments in the lab cannot be possible because of the cost or time. So what we do is that we are using computational models and different basically tools such as machine learning or optimization tools in order to make that happen. So I'm going to mostly talk about one of the projects, which is within EBICS and in collaboration with Melissa Kemp's group from Georgia Tech, which studies patterning in stem cells. But the proposed approach can actually be used in any kind of system. And we also had like some results with Todd McDavid's group earlier. Like as you all know, like people working in EBICS, we know that the spatial and spatial temporal control of patterning is a first step in order to do the engineering of the human organoids and tissues. And what we're trying to solve is that if we have a system with some tunable parameters, how we can actually get a desired pattern. So we want to like see, for instance, for the stem cells, where we start with a population of pluripotent stem cells, and making some changes in the experimental, we start the differentiation. How, like, what changes we need to apply to the system, or what experimental conditions we need to satisfy in order to get some specific pattern, and also at a specific time. And the way that we formulate this problem is through optimization. So we think of this problem as an optimization in which we can use machine learning techniques for basically defining what our patterns or like the desired patterns are. And we are also using some logic called temporal logic, which allows us to also add constraints on the timing of the patterns. And basically what we need to start our work is a computational model, which we usually get from our collaborators. And then we start by providing the data for the machine learning tool and the temporal logic, and also like formulating or solving the optimization. So again, like this is one work with Melissa Kemp's group, and it's not working, but basically this is like an agent-based model of differentiation in cells. And this is a very simplistic basic equation that describes this model, but it's actually from Chad Glenn's from Melissa Kemp's group, which kind of like specifies the differentiation in stem cells through the changes in the concentration of cyclic AMP. And there are some parameters in his models, such as the production rate and the degradation rate, which are the tunable parameters. So he can actually change these parameters within lab. And our work was basically how we can find what tunable parameters can lead to a specific sequence of patterns. So again, like what we need to start our work is a computational model and data for the classification. We can do the classification in two different ways. We can use image-based classifications, which we need to collect data or images of the data. For instance, in the stem cell systems, we had already like classified the patterns that would emerge through the experiments into 8 different patterns. So now using this like a simulator or a computational model that we have, we can basically generate different patterns with different colony and shapes and sizes, and generate the data of these patterns. Or is basically using the feature-based classification, which instead of working with images, which may require a large data set, we can actually extract some meaningful features from the system. So for instance, if you could just like look at these patterns, you can see that we can classify them, or like you can distinguish these patterns based on like the ratio of the differentiated or undifferentiated cells, or whether they are differentiating inside, outside, and other features that can be basically found and like validated through data analysis approaches. So like both of these work. So as soon as we have the data, either images or either features, we can basically use classifiers or train classifiers for our desired pattern. So for instance, if we want to basically reach an outside pattern, which is shown like here, which is like a pattern similar to this one, we can start by training a classifier by considering this as the positive or like a desired class, and all the other patterns as our negative classes. And this will allow us to having some spatial scores that can be used to measure the similarity of the pattern that we may see with the desired pattern. So for instance, if we like train classifiers for this desired pattern, which is the outside pattern, now if we test the classifier against any of these two basic images here, we will get a positive score, meaning that this is similar enough to our training data set. And if it's compared to any other patterns, like either it's random or any other shape of patterns which is not outside, we get a negative score. And we can also combine this score, which is only spatial, to temporal score, which is like if we have basically requirements to be satisfied within specific time points. For instance, we want to reach the outside pattern in the first 2 days of the experiment. We can also specify it, and we can also use basically like look at this classifiers or like spatial scores over time in order to get a spatial temporal score. So for instance, if we run the model for some set of tunable parameters and we get this trajectory, we would get a positive score, which means that this basically transition in the cells is similar to what we had assigned or what was desired for us. Whereas if we get the trajectory or the set of results from the experiments over the second set of tunable parameters, we may get a negative score. And because of this basically a score, we can use optimization. Instead of doing just trial and error or just like looking by eyes, we can have an automated framework or optimization formulation, which basically based on our on trained classifiers, we can automatically generate different trajectories for different set of tunable parameters and try to maximize the similarity score until we get the desired pattern. And we have actually used this framework to try to synthesize both the spatial patterns and also sequences of patterns over time. For instance, this is the result that we had for reaching an inside pattern where like cells start to differentiate from the inside within the first 10 hours of the experiment, and also like a sequence of patterns changing from globular differentiation to snake or like connected differentiation, and also like inverse snake differentiation. And these are all done in basically like the simulation. We haven't validated the results yet, but again, the same idea and same framework had been already used in our lab by DeMarcus from our lab, and also Ashley from Todd's group. And they actually had the validated experimental results where they could synthesize bullseye patterns both in silico and also in vitro. So I guess the challenges of our work is that, first of all, we need an accurate computational model, which exactly mimics the experiments that is done in the lab. So most of the work is usually, well, like most of the time is usually spent on making sure that the data that we have from the experiments is or the capture that data. And we definitely, for the classifiers, would need a good data set of patterns. And also, because most of the models are like already expensive, like for instance, for the patterning model from Melissa's group, it would take 2 hours to only test the results of the experiments for just like one set of tunable parameters. And we need to kind of look for thousands number of tunable parameters. We would need computational powers, which we have at BU. And these are the collaborators that work with our lab, and also we are having other projects with Round-based group as well. And that's it. Thank you.
Thank you.
Thank you.
What are your thoughts about nesting these? So, being able to come up with a classifier that is relevant for the entire, set of cells,